The overall goal of this procedure is to measure the flow of cytoplasm during wound healing in a single cell. This is accomplished by first collecting live Stentor Coeruleus cells to use as a model system for cellular wounding. The second step is to prepare the glass tools for cutting the cells. Next, the Stentor Coeruleus cells are cut open on a customized microscope setup that allows for simultaneous low magnification surgery and high-resolution imaging. The final step is to perform computational analysis of the cytoplasmic flow using particle image velocimetry software. Ultimately, high-resolution imaging of the cytoplasm is used to show the active response of cells during wound healing.
This method can be used to address key questions about cellular wound healing, such as whether or not the cytoplasm becomes gel-like around the region of a cut in order to prevent leakage into the surrounding medium.
Visual demonstration of this method is critical, as the cell handling, tool preparation, and cutting steps are difficult to learn, as they require manual dexterity.
To begin, obtain Stentor Coeruleus cells from either natural ponds, as described by Tartar, or from a commercial supplier. Add 300 milliliters of modified Stentor medium adapted from recipes by Tartar and Deterra as described in the accompanying text protocol. Maintain cultures by growing them in the dark at 20 degrees Celsius. Next, grow up cultures of Chlamydomonas reinhardti algae in tris acetate phosphate medium, using standard culture methods. Prior to feeding, wash the Chlamydomonas cells by gently spinning them down, removing the supernatant, and suspending them in the modified Stentor medium. Feed the 300 milliliter Stentor Coeruleus cultures with about 30 million Chlamydomonas cells.